I'd like to introduce now our next speaker, Carolyn Calfee. Carolyn is a professor of medicine in the Division of Pulmonary and Critical Care Medicine at UC Health, and is doing a tremendous job both as a researcher and as a clinician during this pandemic. So Carolyn, if you can unmute yourself and your video, we'll put you up.
Great. Thanks so much, David. I'm speaking today on behalf of a number of physicians, physician scientists, and bench investigators about the COMET study, which stands for COVID-19 Multi-phenotyping for Effective Therapies. Next slide, please. So the goal of the COMET study is to perform immunophenotyping in order to inform therapeutic development. This project was actually initiated by leadership at the National Institute of Allergy and Infectious Disease and will be coordinated with multiple US sites. We'll be performing host immunophenotyping to identify features that are predictive of COVID-19 severity or progression, with the ultimate goal being to inform development of novel therapies. And we're working on a very quick startup timeline for obvious reasons. We aim to start enrolling within several days to a week. The patient population we'll be studying is adults that are hospitalized at Moffitt Long or San Francisco General for COVID-19. And this work builds on a well-established ICU cohort that's been operational for over a decade called the EARLY cohort, which stands for Early Assessment of Renal and Lung Injury. And we'll be obtaining serial blood and tracheal aspirate samples with comprehensive immunophenotyping and a rich clinical database. Next slide. So you've already heard from Chaz about some of the molecular phenotyping work that we've done in this cohort in the past. I also wanted to just briefly mention that we also have a history of doing a lot of work with clinical data. This is a paper that was actually published just this week in Intensive Care Medicine, led by Katie Oriema, who is a former UCSF chief resident, on acute respiratory distress syndrome attributable mortality in critically ill patients with sepsis. For this project, we enrolled over 1,000 patients in the ICUs at Moffitt Long Hospital and San Francisco General, hand-curated sepsis and ARDS diagnoses for all of them, and identified that ARDS had an attributable mortality of 27%, which was primarily driven by severe disease, as you can see at the bottom figure. So this is just to emphasize that we intend for this project to have a very rich clinical data set in addition to the rich biological data set that I'm about to tell you about. Next slide, please. So the primary hypothesis of the COMET study is that immunophenotypic characterization of COVID-19 patients will identify biologic predictors of disease severity and clinical outcomes, and that these predictors may then suggest candidate targets for novel therapeutics and subsets of individuals who are most likely to benefit from them. Next slide. This slide gives you an overview of the process that we're going to be pursuing. So on the left-hand side, you see the sampling pipeline, which focuses on COVID-19 positive inpatients sampled throughout the course of disease, including as important controls, some COVID suspects who test negative. We'll be doing longitudinal sampling with the schedule that you see listed on the screen below throughout hospitalization and, importantly, in convalescence. And for patients who are transferred to the ICU, we'll have increased sampling frequency over the first 48 hours they're there. The primary assays we're planning at this point are shown on the right-hand side of the screen. And these include a variety of different assays in the blood, including serology, transcriptomics, T and B cell characterization, cytoph and flow, plasma and serum proteomics, as well as others. In addition, we'll be doing studies analogous to those that Chaz showed you in the tracheal aspirate, like metagenomic sequencing, as well as viral load. Also going to be doing some novel UCSF assays at the Chan Zuckerberg Biohub, as you can see at the bottom of the screen. Next slide, please. So as we all know, there are special challenges to doing this type of clinical research during a pandemic. And that's why I think now more than ever, it's really important for us to emphasize cooperation and collaboration. And so we've been working to try to unite researchers across UCSF to build a rich clinical and biological data set that maximizes inclusivity and teamwork and coordinates with both NIAID and with the NHLBI. I can't emphasize enough, as somebody who's been working with critically ill patients for a long time, how important it is to minimize the burden on patients, families, and bedside staff who at this time are particularly under considerable stress. And so coordinating our efforts is particularly important. We'll be doing a contactless phone consent in order to protect patients and staff and conserve vital personal protective equipment. And we'll also be coordinating with other COVID-19 related studies. And I want to take a moment here just to highlight a couple of those. One is the NHLBI's Prevention and Early Treatment of Acute Lung Injury, or PEDAL network. This is a clinical trials network focused on ARDS that we've been part of at UCSF here for decades, led by Michael Mathey. In addition to their clinical trials work, they're planning an observational cohort with clinical and biologic data focused on ARDS, and we'll be coordinating with these efforts. We'll also be coordinating with the ICERIC registry, which stands for the International Severe Acute Respiratory and Emerging Infection Consortium. In addition, we're coordinating with clinical trials. I want to mention one in particular, which is the Department of Defense funded STAT trial. This is a clinical trial led by Michael Mathey of mesenchymal stem or stromal cells for ARDS. It's actively enrolling now, and in fact has already treated a patient with COVID-19 related ARDS. And then Dr. Lukemeyer will be talking later about the ASTR trial and remdesivir trials. Next slide, please. So I want to echo what you've heard from, I think, all of the previous speakers, which is to say that it's been incredibly inspiring to see how quickly and selflessly and generously people from across UCSF have come together in this setting. NIAID approached UCSF last week about this study, and over the past week we've had over 50 investigators, postdoctoral fellows, and staff, not all of whom I could even fit on the slide, come together to work hard on getting this study up and off the ground. I want to emphasize that this is a collaborative project, including UCSF Health, ZSFG, the ImmunoX Group, UCSF CoLabs, and the Chan Zuckerberg BioHub, and includes all of the departments and divisions shown here, as well as many others. Next slide, please. So with that, I'll close, and I'll just say that we welcome your engagement and contribution. If you have interest in contributing or additional questions, please feel free to contact Alyssa Ward, who's one of our project managers, or you can also contact me directly. Thanks.